Our next little vignette, if you will, is going to be looking at energy and metabolism. How do you get things done? And this is where we're going to go back to proteins. We're going to go back to enzymes. So I want you to, have that in your mind. How do you make things happen? Energy and metabolism, how things work. Energy, what is it? It's the ability to move or change matter. That's the official definition. But you can think of somebody who you would say has a lot of energy or doesn't have a lot of energy. We need to consider the law of conservation of mass at this point, and that states that matter can neither be created or destroyed. It just changes form. Similarly, in the law of conservation of energy, the total amount of energy does not change. It may change from one form to another. Chemical reactions are important at this point because this is how molecules come together, how they go away, break into smaller units. When we were looking at monosaccharides or monomers, disaccharides, how do they form those and how do they break down? A chemical reaction is required to change a substance. Whatever is coming together to react or whatever is being reacted upon is called the reactant. The product is what we get at the end. And if you notice, the arrow goes both ways in this, so that means that products can become reactants again. The reaction can go both ways. Activation energy is important to consider. This is the minimum amount of energy required to make a reaction happen. Why do we care about this? Well, I want you to think about, in order for a reaction to happen between two molecules, they have to actually come together. And in order for molecules to come together, they often have to move faster. They have to need more energy. Think of all of yourselves in the gymnasium. If I put 4 of you blindfolded in the gym and made you walk around until you reacted or bumped into each other, how long do you think it might take? Probably a while. But if I made you run, would it go faster? Would you be more likely to bump into each other? That's activation energy. Well, enzymes are something that we're going to be talking about now. They're very important because they help reactions occur faster. Without them, reactions really would take forever. Enzymes lower the activation energy. So take my scenario with you blindfolded running in the gym. Imagine if I held one of you still, and that I was acting as the enzyme. I was holding you in place to make it easier for you to react with the other guys that are blindfolded running around the gym. It will actually happen. You don't have to run as fast because it's going to happen. You're going to react more quickly. Enzymes are not changed in a reaction. They stay the same. Only the reactants are changed. And enzymes are very specific for a given reaction. So an enzyme for sugars will not work on proteins. Here's a diagram of an enzyme. The ligand, the yellow thing, that's what is going to fit in the enzyme and whatever molecule that is. It can also be called the substrate. The protein or enzyme is the big blue globby thing, and I want you to think about when the paper towel. Remember I folded that paper towel up into a big globby thing? That's what gave it its shape. So you notice that the enzyme has a unique shape that matches the unique shape of the substrate. Active site is the place on an enzyme where a molecule binds. It's that funny little unique shape that is called the active site, and that's where the substrate will bind. The substrate is in blue here, the enzyme in yellow. The substrate is the molecule that binds to the active site and will be changed in the reaction. And again, the enzyme itself is not changed. So if you look at the diagram, we start on the left, we have the enzyme and the substrate. Then we have the enzyme substrate complex. And in this case, the enzyme is causing the substrate to break into 2 products. And you notice that the enzyme didn't change at all on the far right. Now, there's a number of factors that can affect enzyme reactions. Temperature can make molecules move faster and interact more. However, too high of a temperature can denature or destroy the enzyme. pH is also something that can vary and can affect the enzyme reaction. Each reaction has an optimal or a best pH. The other thing that can other kind of factor that can affect your enzyme reaction would be the concentration of either the substrate or the enzyme. Okay, remember the paper towel? I just mentioned it a minute ago to recall that this is our protein in its tertiary structure. And you'll notice it looks like a big lumpy, clumpy thing. But right in here is my active site. And here's my substrate. An ordinary tennis ball, you say? Well, I say it's substrate. And it fits very nicely into my active site. Now, a moment ago I mentioned that temperature, okay, and even pH can affect the enzyme activity. And one thing that can happen, if we get the temperature too high, I said it could denature or destroy that enzyme. What does denaturing mean? Well, what it will do is break all those bonds that were holding that enzyme together, and oops, where's the active site? It's disappeared. Okay, we've gone back down to our primary structure. No more active site, no more reaction.